Good day, my dear students. Welcome once again to lecture series 3 of Biochemical Pharmacology, BCH 407. The topic for today is molecular basis of chemotherapy. Now, before we proceed, let's look at the focus of this topic. They are 1. Definition of chemotherapy, drug action on DNA, drug interaction with lipid molecules, drug-enzyme interaction, mode of drug action on calcium channels, and drug interaction with ion pump enzymes. Introduction. With the exception of very few organisms, others have the following molecules in their body system. 1. DNA or RNA. Secondly, you find lipid molecules, you find enzymes, you have ions, particularly calcium ion channels, and ion pump enzymes. These are biomolecules of immense importance. Moving forward, how do we define chemotherapy? Chemotherapy is a term used primarily to refer to the treatment of infectious diseases and cancer, or the use of cytotoxic substances in the treatment of infectious diseases or cancer. Those two definitions mean the same thing. So anyone you can easily recall will answer the question. Now let's look at differentiation in definition. Now, chemotherapy, when applied for the treatment of cancer, is referred to as cancer chemotherapy. On the other hand, when used for the treatment of microbial infection, it is called antimicrobial chemotherapy. Now, what is the action of drug on DNA? But before we go into that, let's look at the key biological functions of DNA, which are 1. Storing and transmitting genetic information. 2. Control or detection of protein synthesis. 3. Ensuring accurate cell division and inheritance. A deviation from these 3 precise functions will culminate in a disease state, one way or the other. You must have heard of the term cell cycle. Cell cycle is a series of events that takes place within a cell. The event results in the duplication of DNA and division of the cytoplasm and organelles to produce 2 daughter cells. Now, the cell cycle consists of 4 phases, namely the G1 phase, the S phase, and the G2 and the M phase. Of course, we have the GO phase, which is the resting phase. Each phase has distinct events, and they are critical for cell growth, DNA replication, and cell division. Now, let's look at the phases of the cell cycle and their events. Starting with the G1 phase or the gap phase 1. In this phase, cell grows in size and increases in its metabolic activity. It synthesizes messenger RNA and proteins necessary for DNA replication in the next phase, and then assesses signal to determine whether to proceed to S phase or enter the dominant phase GO, which is the resting phase. These are the sequences of events in this phase. Now, the S phase. The S stands for synthesis. Synthesis of what? Synthesis of DNA. This is where DNA replication occurs after the synthesis of mRNA. It is also where histone proteins are synthesized to package the newly replicated DNA. Now, gap phase 2. Under this phase, the cell continues to grow and synthesizes proteins needed for mitosis. It screens for DNA damage or incomplete replication to ensure correct genomic is produced before entering mitosis. Now, I must emphasize that a deviation from these precise functions or this precise event in the cell cycle, as I said earlier, will result in a disease state. Non-covalent binding, covalent binding, and DNA cleavage. Consequences. What are the consequences of these actions? These interactions can have severe effects on the cell cycle, such as DNA replication, cell division, photosynthesis, and DNA repair mechanisms. Now, what does this mean? Having a severe consequences on this means either DNA cannot be replicated, cell cannot divide, photosynthesis may be halted or inhibited or faulted. DNA repair mechanisms may be perturbed. In any of these events, the result is cell death or cell cycle arrest. Now, when these events are occurring, it is not occurring on the individuals. Remember that the drug you are taking or the drug you have taken is not meant to kill you, but to deal with the microbial infection that has occurred in your system. So it's a fight against the microbes, which also have their life or their cell cycle. So the drug will interact with the cell cycle of the microbes and not your cells. Please note. Now, drug interaction with lipid molecules. Cell membranes often constitute the first biological structure encountered by drugs. You can see here the structure of the lipid bilayer, the components, glycolipids, glycoprotein, you have proteins, some embedded, some transversing, that is integral, and some surface proteins. Then you have the cholesterol, you have the channels, then you can see the phospholipid bilayer. Now, two types of drugs, namely hydrophobic and hydrophilic drugs, interact differently with membrane lipids, influencing structure, function, and dynamics. Of course, you know that for a healthy individual, for a healthy system, the integrity of the lipid bilayer must be preserved. Again, any alteration from there in terms of structure and dynamics will certainly affect its function. Now, hydrophobic drugs interact with lipid molecules by one, partitioning into membrane lipids. By this, they preferentially integrate into the hydrophobic core of membranes. When this is done, it now affects membrane fluidity and packing density, depending on the drug lipophilicity. Second, hydrophobic drugs interact and affect membrane perturbation by causing changes in lipid organization, such as condensing effects on a side chain or creating voids within the hydrophobic core. Such perturbations can impact on membrane permeability and protein function. In the same way, the hydrophilic drugs have the effect of limited membrane penetration. Hydrophilic drugs generally do not integrate well into the hydrophobic lipid membranes due to their polarity. Of course, they also have limited membrane, like I said earlier, limited membrane penetration, in that their incorporation into membranes often require modifications, such as attaching hydrophobic group or using specialized carrier like liposomes. Hydrophobic drugs can influence membrane fluidity indirectly by interacting with polar head groups or inducing changes in lipid composition. Such changes may be by way of membrane thickness and elasticity, which is the same as fluidity. Now, let's quickly take a look at the membrane thickness and elasticity. Drugs can influence membrane thickness and elasticity by altering the lipid composition, protein interaction, or cholesterol content in the membrane. Now let's look at alteration of lipid composition in the lipid bilayers. First, insertion of longer chain lipids increases membrane thickness. Secondly, shorter or unsaturated lipids can reduce thickness of the membrane. Thirdly, some drugs influence cholesterol levels in membrane. Of course, cholesterol packs tightly with phospholipids. More cholesterol often leads to thicker and more ordered membranes. Now let's look at drug-enzyme interactions. Most drugs' action on enzymes may be either by enzyme inhibition, enzyme induction, activation, or suppression. In enzyme inhibition, there is a reduction or stoppage of enzyme activity, while in induction, there is increased production of certain enzymes. Activation. Certain drugs can enhance the activity of some enzymes. Suppression. This reduces the expression of enzymes at the genetic level, leading to long-term reduction in enzymatic activity. Now we look at all these in turns, starting with enzyme inhibition. There are several types of enzyme inhibition, namely competitive, not competitive, uncompetitive, and irreversible inhibition. In competitive inhibition, the drug competes with the natural substrate for the active site of the enzyme. In non-competitive inhibition, the drug binds to allosteric site of the enzyme, and in uncompetitive inhibition, the drug binds only to the enzyme substrate complex, preventing the reaction from proceeding further. In irreversible reaction, the drugs form a covalent bond with the enzyme, permanently inhibiting it. Remember when we are talking of the bonds in chemical reactions, I told you that covalent bond is undesirable. In enzyme induction, certain drugs can cause an increase in the synthesis of specific cytochrome P50 enzymes, leading to increased metabolism of drugs, reduced drug effectiveness, and potential for drug interaction. In the case of activation, some drugs have the ability to enhance the activity of enzymes, though these are less common but they are important in certain therapeutic settings. In enzyme suppression, we see a situation where there is a decrease in the production or activity of enzyme, usually through the regulation of gene expression or inhibition of protein synthesis. When a drug suppresses an enzyme, it results in lower enzyme levels or reduced enzymatic activity within the cell. Now let's look at calcium channels. They are transmembrane ion proteins that selectively conduct a calcium ion through the cell membrane in response to membrane potential during depolarization. Now, the figure or the picture below or the structure below, you can see you have the components that make up the calcium ion channel within the membrane lipid bilayer. Now let's talk about the description of calcium channel. It is composed of 4 to 5 distinct subunits, as you can see, within the lipid bilayer. The alpha subunit is the largest, and it incorporates the pore region, the voltage sensor, gating apparatus, several channel regulation sites, by-second messengers, drugs, and toxins. There is an intracellular beta subunit, a transmembrane alpha 2 delta subunit complex, and a transmembrane gamma subunit. Now, these are the various types of calcium channels. You have the L type, the T type, N type, PQ type, arrow type, NMDA receptor, and so on and so forth. Each type have its own peculiar function it performs within the cell system. And the location of the various types is also indicated on the table here presented. Now, we talked about ion potential and depolarization. Now, let's quickly take a look at this in order for us to understand or have an insight into how drug affects calcium channels. The key concepts here is on how cells communicate and function. Ion potential is just electric charge difference across a cell membrane. As you can see here in the picture presented, you see the positives and the negative. Ion potential maintains this charge difference by controlling the movement of ions like sodium, potassium, calcium, and chloride through specialized channels in their membranes. Normally, the inside of a cell is more negatively charged than the outside. This state is called the resting membrane potential or polarized cell membrane. Now, from the resting potential, we come to depolarization, which happens when the charge difference across cell membrane become less negative or more positive. This occurs when ion channel open, allowing positively charged ions like sodium or calcium to flow into the cell. In this case of consideration, calcium. Depolarization is a signal that tells the cell to activate or fire, as in the nerve cells. Depolarization triggers electrical signals called action potentials that travels along the nerves. Depolarization, therefore, can lead to muscle cell contractions. There are several types of calcium channels, but the L-type calcium channels are the most significant because of their crucial role in cardiovascular activities. Calcium channels are protein structures in the cell membranes that allow calcium ion to enter cells when triggered. These channels are found in the heart and blood vessels, where they control contractions, in the nerve cells, where they assist in neurotransmission, and in muscles for contraction. Let's look at drug actions on calcium channel. Two types of drug act on calcium channel. The first is calcium ion channel activator and calcium ion channel blockers. The, act, the calcium ion channel enhances calcium influx into the cell, while the blockers inhibit calcium influx. Calcium channel blockers bind to the alpha 1 subunit of L-type calcium channels and prevent calcium ion from entering the cell during depolarization. The consequences of this action is that there is a decreased muscle contraction, reduced cardiac workload, and vasodilation. Drug action on ion pumps. Ion pumps are all transporters that can thermodynamically move ions uphill. They are there are primary and secondary ion pumps. Ion pumps that hydrolyzes ATP are primary pumps. Secondary pumps exploit the energy stored in ion electrochemical gradients by coupling thermodynamically downhill movements of those ions to drive the uphill transport of another substrate. Secondary pumps are sometimes called co-transporters or exchangers, according to the relative directions of the coupled downhill or uphill ion flow or movement. Now, difference between ion channels and pumps. A channel has a single gate, and a pump has at least two gates that should never open at once. Now, what is a gate? It can be considered to be part of the protein that precludes ion movement along the translocation pathway in the prohibitive conformation, but not in the permissive conformation. Now, figure A and B below clearly shows the difference between ion channel and an ion pump. You can see A here has a single gate when closed and when open. Figure B, the ion pump has one gate open and the other gate closed. As you can see from the figures, when the ion channel is permitting a molecule in and out, the ion pump is exchanging ions. Now, what can be said to be the drug action on ion pumps? Drugs may interact with ion pumps through various mechanisms such as activation or inhibition of signaling pathways to bring about significant drug effects. These effects may be positive or negative, depending on the drug. Now, the recommended test is given and it is available at the bookshop. Study questions. I hope that having listened carefully to these lectures and having gone through it with me, you should be able to answer these 6 questions conveniently. This is the end of the lecture. If you have any question, and I believe you should, just note them down. When we meet physically, you can raise your questions and it will be a class discussion so that everybody will benefit from it. Thank you for listening and watching. See you in lecture 4.